Greetings, guys, gals, non-binary pals. Welcome back to the Amazing Plant Project. My name's Kate, and today we're doing part 2 of chapter 3 of the plants. So, let me just see if I can round up some more folks in here, and we'll have at it. Okay. One sec. Alright, yeah, so it's working. And we'll just turn down on that. And there, come on. Okay. Alright, so we've got a... Okay, one sec. Audio. Okay. So I know it's transmitting. Oh, come on with this thing. Alright. And okay. There we go. So where did we leave off? Yesterday? Last week. Honestly, the days just kind of have been running together a bit too much. But one sec. Let me see if I can weight the thing down more. There we go. A little bit less wonky then. Alright. So, we left off on the endoplasmic reticulum, which where'd it go? It's not all that well done in here, but it's this guy up on the side. Alright. One sec. Alright, so here we go. The endoplasmic reticulum is... Let me see if I can... There we go. All kinds of transmission. Hey, guys. Alright, so. How's everybody doing, first off? You doing alright? Okay. So, endoplasmic reticulum, or the ER for short, which, if you're writing it out, makes for interesting things in the logistics, depending on context. But the endoplasmic reticulum is it's a complex, like, three-dimensional membrane system. Oh, hey, Sonny! Welcome to the chat and fun things. Alright. All kinds of fun. Alright, so 3D membrane system that permeates the cytosol, and we know from last week that the cytosol is the for lack of a better term, the gooey interior of the cell and all the things in it. So there's that. In a sectional, so again with the water. It's been 5 minutes. And apparently there's hair everywhere, honestly. But in a sectional, so like a cross-section view, and that's usually how it's described when you're looking at the diagrams in a textbook. It's like a section so you can see all the bits. But in a sectional view, the endoplasmic reticulum appears as two parallel membranes with a narrow space or lumen between them. And the type or amount of the ER depends on the type of cell, the metabolic activity of the cell, and the stage of development of the cell. So a rough ER, so that's the rough ER is the one that has the ribosomes on it. Is more present in cells that store proteins, and the flattened sacs or cisterna, which cistern is a holding container, so that's where the term would have come from. Just like most things, it's largely Latin and Greek as the origin story of stuff. But the cisternae, or cisterna for the singular, have numerous polysomes. So the polysomes were the conglomerates of rhizomes from last time. Yeah. That would be where the protein synthesis is occurring. And so it's got more or numerous polysomes on the surface versus the smooth ER that is more present in cells that are secreting lipids or oils generally. And they lack the ribosomes, or rhizomes. Yeah, they lack the rhizomes. And it's largely kind of a tubular formation. And that's involved in the lipid synthesis. So we've got the rough ER that is synthesizing the proteins, and the smooth ER that is synthesizing the lipids generally. I thought that's what it meant, yeah. I mean, honestly, that would be more straightforward, but it's not as specific sounding. Yeah. Yeah, basically. That fits, though. And both of those exist in cells, and they're constantly in flux. They're constantly changing and moving around, just like everything else in the cell structure. Then we've got the cortical. I might not be saying it right. Cortisol? Cortical. We're going with cortical. But it's the cell's interior outer membrane. So, like, right along here. Come on. Yeah, okay. Where was I? That's like just inside the cell wall on the very peripheral of the ectoplasm. That's the cytoplasm. Jesus. Uh like when I read stuff, it gets like the correlations and whatnot sometimes transmit a bit too much. And apparently I've had ghosts on the brain more than usual. Right. Yeah. That yeah. That's what I was thinking, because I don't think there's two that sound pretty much the same. So I'm guessing cortical is the more accurate pronunciation on that one. Um, don't think there's a correlation with those two. But yeah, cortisol's the stress, so uh, but yeah, so peripheral cytoplasm. There we go. Um so we've got the cortical endoplasmic reticulum that appears to kind of regulate the level of calcium ions in the cytosol. So that would be Ca Ca2 plus. Yeah, ghost in the cell. Because puns and I can. Huh. Exactly. Um, but the cortical ER might possibly play a role in other physiological and developmental processes involving calcium. I mean, I have many nerdy fandoms that I'm a part of, so all of them happening right here. All of them. Anyway, so might play a role in other physiological or developmental processes involving calcium. And it's also been suggested that the cortical ER kind of stabilizes or anchors the exoskeleton of the cell. I just follow it on here. Yeah. Okay. and it's a general indicator of the metabolic and developmental status of said cell. The more static or quiescent the cells are, the less they have generally. And developing or active cells tend to have more. Sometimes the ER appears to be kind of a continuous portion of the nuclear envelope, so the membrane around the nucleus that protects it. And often the nuclear envelope is considered an extension of the ER. Interestingly, during cell division, the new nuclear envelope is formed from parts of the former endoplasmic reticulum, because in cell division, the nucleus is kind of ripped apart in order to separate the chromosomes and create the daughter cells, which we'll get to in a little bit, but something to keep in the back of your head as we're going. And it functions as kind of a communication relay system. And what did I write? Channels. There we go. When you can't read your notes, it's kind of bad. And channels needed materials to different parts of the cell. So examples, proteins and lipids. Yeah. Just recycling all the bits, which is nice. And yeah, so the ER of adjacent cells is connected via the plasmodesmata, or the what's the term? I can't remember off the top of my head. It's bugging me now. Okay, one sec. Oh, yeah. Back to the source material real quick. What is that? It's one of those, I totally don't need to write this down. It's fine. No. I don't need to remember that, because I already have it in my brain. Now I can't remember it. What am I even looking up? Oh. Aha. Here we go. Okay. The cytoplasmic threads. Which are these guys? Right here. Okay. Now that we've gotten that sorted out. Very true. Very true indeed. Okay. So, what was I doing? Alright. So, the endoplasmic reticulum of adjacent cells is connected via the plasmodesmata. That's the cytoplasmic strands. And it aids cell-to-cell communication. And that tends to be in, as I whack myself in the face, tends to be via the primary pit fields over here. Alright, so next on the list here, we've got oil bodies. So the ER is one of the main lipid synthesis sites. So it produces the lipids, what would become the oils and fats and waxes and stuff. And the lipid droplets arise in the ER and are released into the cytosol. So they are formed in the endoplasmic reticulum and are then disseminated to the rest of the cell. Yeah. They're kind of spherical structures. Just think like a drop of oil in water, it kind of balls up because it's a droplet. Same kind of principle. And it gives the cytoplasm a kind of granular appearance when you look at it under a light microscope, because there's a bunch of different droplets in there and the light is refracting off of them, so it looks kind of like sand. It's most abundant in fruits and seeds, and about 45% of seeds is oil. Like the contents of the seed is oil, and it's an energy source and a source of carbon to the developing seedling, which is why if you have seeds or nuts, a lot of times it's recommended that you keep them in a refrigerated or at least a cool space, because the oil in them can go off relatively easily, and you don't want to eat that. You really don't want to eat that. Interestingly, though, if you're doing stuff with oil, this is completely not in the text at all, it's just random info. If you're doing stuff with oil, like you're making a perfume or something, you want a carrier oil to hold it, but you don't want the oil to go bad. Fractionated coconut oil doesn't go off. So if you use that, the whole setup might go a bit wonky eventually, but the oil won't go bad. And you can keep it at room temperature and it's fine. Versus most other oils that have a kind of short to medium length life expectancy before they kind of go wonky. Yeah. Yep, fractionated. If I can find it on like Amazon or something, I'll link something in the description after this. But I think I got mine at Smart and Final. So just the grocery store. So we shall see. Oh, that one. But the oil acts as an extra energy source for the seedling. That's why a lot of times if you're doing oil prep, it's the seed itself that you're grinding in order to get the oil out. Sometimes it's the leaves, sometimes it's the flowers, but a lot of times it's the oil because that's where the highest concentration is. It also depends on the properties that you're going for as to what kind of oil and the time of year and the time of day and everything else that you need it and yada yada. I think for the fractionated bit, it's just I have to look at mine. I think it's that they've distilled it in a certain way, but that doesn't make sense because distilling wouldn't work with oil as much. I mean, it would, but not entirely sure. But yeah, I have a phone. I can look it up. And we shall see. And now we see the research process in its natural habitat. Okay. Da, 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 da. Yeah, okay. Fractionated coconut oil is virgin coconut oil that has been distilled, allowing long chain fatty acids to separate and solidify for removal. Da, 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 da. What remains is an ultralight, odorless coconut oil that stays liquid in form at cooler temperatures. It's more easily absorbed by the skin and less likely to clog floors. Less likely to clog pores than virgin coconut oil. It also maintains a longer shelf life, nourishing medium chain fatty acids and essential nutrients in this form. Yeah. Yeah. So it's distilled, essentially. Huh. I know the feathers, just for birds in general, feathers need to maintain an appropriate oil concentration in order to maintain their structure and be waterproof, water resistant, and groomed properly. It probably has something, like some underlying element that makes it more shiny for the hummingbird feathers. But you see the shine refraction even after the feathers have been removed, like years after. And unless the feathers have just like remained having those oils, I don't know that it would make that much of a difference. Just because if you go to museums and stuff and look at there's cloaks and headdresses and stuff that I've seen from native peoples, and some of them have hummingbird feathers in them, or peacock or what have you, and they all retain the same kind of almost multi-chrome shine that is indicative of that species, but they haven't been attached to a bird in a good 50 to 100 years sometimes, if not longer. So, yeah. Wait, air bubbles? Really? That's cool. Okay. But, like, they're... What? Backdrop's trying to come off the wall. Alright. That is really cool. Alright. Yay for learning stuff. Okay. Stop it. I have to tape that back up. Alright. Okay. What was I doing? Developing seeds. Okay. Here we go. So now we've got the Golgi apparatus. That is a whole thing unto itself, just like the Endoplasmic Reticulum. Alright. So the Golgi apparatus refers to the conglomeration that is all the Golgi bodies, or the dictosomes, in a cell. And it's stacks of flattened, disc-shaped sacs, or the cisterns, often branched into tubules at their margins, or the endpoints. Right? It's being so rude, honestly. See? That's it, behave. I'm watching you. Stay. Alright. Anyway. So it's a dynamic and highly polarized membrane system that has two faces. So the forming face tends to be on the interior, facing the endoplasmic reticulum, and the maturing face tends to be on the exterior towards the outer part of the cell. So basically, like, the endoplasmic reticulum makes things, sends it to the Golgi apparatus, it goes through that, and then is dealt with. Sometimes. Sometimes it's more roundabout than that, but you know. For the things that go through there, that's generally what happens. So the trans-Golgi network is a structurally biochemically, that's what that says, a structurally and biochemically distinct compartment of the Golgi apparatus. Component of the Golgi apparatus that exists on the maturing phase of the Golgi body. So we've got forming, maturing, and it's involved in secretion. So it's most involved in synthesis and secretion of non-cellulosic, that's a big word, cellulosic polysaccharides that are to be incorporated into the cell wall. So polysaccharides that are not cellulose, that are going to be put into the cell wall. And also the process and secretion of glycoproteins that are transmitted via the rough ER. Yeah. So, that's two. Okay. Via the rough ER to the transmission vesicles that would be transmitting the elements. So if this is the Golgi apparatus, let's say that's the Golgi apparatus, right? It's like distinct bits. The shuttle vesicles are like the individual pieces, the individual lines going through it, essentially, the sections. And they're sort of step structures in the Golgi body, and it's how the glycoproteins move from the forming to the maturing phase. So they start here and get transmitted here, and then here, and then here, and then are on the maturing side and can do what they're going to do. So it can process polysaccharides and glycoproteins simultaneously. So multifunctionality and stuff with that. And then we've got the coated vesicles that are coated with several proteins, including clathmarin, which I have to say is giving me weird flashbacks to people not being able to pronounce my name. So, yeah. Just put that out there. But, and then we've got the movement of vesicles from the trans-Golgi network to the plasma membrane appears to depend on the presence of actin filaments, which are strands of material that vesicles fuse to, and fuse with. There we go. Yeah. And allows the vesicle to discharge its contents into the plasma membrane. So, like, if, like, so this is the Golgi. If there's a vesicle here, there's an actin filament, and it kind of helps it get over to the cell wall and diffuse its contents into the plasma membrane, either for use by the cell or more likely to be shared with other cells if it's going to the plasma membrane. And we've got exocytosis, which the secretion of substances from cells of material in vesicles. So again, vesicle secreting material. The endomembrane system is also important. That would be all the cellular membranes minus the mitochondrial, plastid, and peroxisomal membranes. The ER is the initial source for the membranes, and it's transmitted through the trans-Golgi body. And it can turn endoplasmic reticulum membrane into tonoplast and I can't read what I put on there, honestly, with this thing. This is what I get when I write fast, I guess. Okay. Plastic. Plasma, there we go. Okay. Tonoplast and plasma membrane-like membranes. Alright. Past that. So, now we've got the cytoskeleton that is present in all eukaryotic cells. It's a network of proteins and filaments that extends throughout the cytosol and is involved with many of the processes, including cell division, growth, and differentiation of the cells, and organelle movement within the cell. So moving the stuff around where it needs to be. We've got microtubules that are cylindrical structures about 24 nanometers in diameter and of varying lengths. They're made up of tubulin that it's subunits. Yeah, they're made up of tubulin. The subunits are kind of arranged into a helix formation of 13 rows or protofilaments around a core, and all the subunits are oriented in the same direction in the profilament. So they all have the same kind of polarity. It's a polar element. Not element, but it's a polar thing. So one end grows faster, and that's the positive end, and one end grows slower, and that's the negative end. And then we've got the dynamic instability. Unintentionally dramatic on that one. Let's try that again. Dynamic instability, where it's in constant flux, essentially. The microtubules in a cell are constantly changing and constantly moving, and there's no like static state for that part of the cell. Oh man. For some reason my nose only gets really itchy when I'm doing streams. I don't know. One of those, yeah. You shouldn't be scratching your nose right now, so like it's fucking me. Alright. And with that, we've got microtubule organization centers that are nucleating sites. So it's the surface of the nucleus and portions of the cortical cytoplasm. And the microtubules also have a role in enlarging and differentiating cells. And the growth of the cell wall is a big part of that. And it controls the cellulose microfibrils. The microfibril alignment, there we go, as it's added to the cell wall, because if the fibers aren't laid out properly, then everything goes all kinds of wonky, and you don't want that. Nobody wants it. They also make up the spindle fibers that play a role in chromosome movement and cell plate formation in dividing cells. So when the chromosomes are separating, there's spindle fibers that kind of latch on to one set of chromosomes and the other set of chromosomes and pull it apart, and then when everything is resituating into the new cells, it's putting it together. So microtubules. And then we've got actin filaments that are microfilaments, also polar structures with distinct plus and minus ends, or positive and negative ends, however you want to say it. It's basically the same thing. But they're long filaments, usually 5 to 7 nanometers in diameter, and are involved in cell wall deposition tip growth of pollen tubes. So when the pollen tubes are developing, it's part of that. Movement of the nucleus before and following cell division. Organelle movement. Vesicle-mediated secretion, because the vesicles are like the kind of the interior lakes in the cell, holding like nutrients and stuff. And organization of the ER and cytoplastic streaming. So, again, moving stuff around in the cell. And the Flagella and Cilia. Cilia. Yeah. Or the singular flagellum and cilium. Almost invariably, there's more than one, so it's generally flagella and cilia. So they're hair-like structures extending from the surface of many different types of eukaryotic cells. They're relatively thin and have a kind of consistent diameter, about 0.2 micrometers wide. It can be anywhere from 2 to 150 micrometers long, depending on the species and what it does and where it is, and various other conditions. Longer structures tend to be present either solo or in smaller numbers, and are generally considered or generally designated as flagella. Shorter structures in greater numbers are generally considered or called cilia, but they're essentially the same thing, and in the text, they're all called the same thing because they do the same thing, they operate the same way. It's just based on the species and what that particular species needs in their environment. They've adapted a certain number or a certain amount of them and stuff. It's the same as a giraffe has an insanely long neck. Humans have short neck. It's still a neck. It does the same thing. It's just one has one that's real long and one has one that's not. So, yeah, same basic principle. Most often, they're mobility structures. So, a lot of times you see that in more aquatic environments or spaces where there's like a contained body of water sort of thing, or a body of like a suspension fluid sort of thing. And the flagella or cilia let different components move around in there. So for cells, sometimes there's like a let's say a symbiotic relationship between a plant and a bacterium or something. It'll be able to move around in there. And other such things of that nature. It's kind of the same with excuse me. It's the same sort of principle for like gut bacteria and all the rest of it. But yeah, so Oh, yeah. Interesting point on those. The movement properties are in the limb itself. It's not in the rest of the cell. So scientists have done studies, and apparently if you remove the appendage from the rest of the cell, it still continues moving because all the momentum and component bits that make it move are in the limb itself, not on the rest of it, which is an intriguing idea, I will say. Now we get to the main thing for what makes a plant cell distinctly different from the rest of the cells and things of that nature. So, the cell wall, which touched on last time, but again, this dude around the edge there. And the difference between plant and animal cells is that for plant cells, the cell wall constrains the expansion of the cell. It constrains the expansion of the protoplast and prevents rupture of the plasma membrane when the protoplast, so the cell, expands upon absorbing water, which is kind of a necessary thing because plants only need so much water, and if you overwater them, they don't like it. There's some that have a more violent reaction than others, like cacti, that if you overwater them, they get real easy. And if you overwater them past a certain point, they can explode. Usually it takes a lot to do that. But if the tension in the cells builds up enough that that's a possibility, it's a possibility. So, something to keep in mind. Also, if you, let's say, I'm most familiar with barrel cacti, just because if you've been on the channel for any length of time, you might have seen Cactus Bob make a few appearances. He's a barrel cactus. Or like a little barrel cactus. A little golden barrel. But when they are lacking water, the spines are really pronounced. Like the ridges are really pronounced. And when they've taken on water, then it's fuller. And if you have a cactus that's nice and full and you water it, it might start to like sort of shrink up a bit. That's because there's too much water in there, which is a weird concept because cacti that have too much water and cacti that have too little water look really similar sometimes. At which point you have to remember whether or not you watered it, and when, and what kind of conditions it's been in since then, because sometimes sometimes it didn't go too well. But they also tend to be fine if you just kind of leave them sit somewhere that's mostly sunny and give them a spritz every once in a while. So. Also, we're getting into summer now, at least in the northern hemisphere. So keep an eye on them. You might need to spritz them or give them a drink of water like every couple of weeks or something. In the winter, don't do that because they're like having a snooze and they're fine. They've taken up all the water that they need and they're good. But during the summer, yeah. So, anyway, back to cell walls. Okay. So it contains enzymes and that plays roles in absorption, transport, and secretion of substances in the plants. It may also play a role in defense against bacterial and fungal pathogens through transmitting the info to other parts of the cell to make phytotoxins. Yeah. To make phytotoxins that are essentially in-house antibiotics or antibodies. Which it's kind of the same principle as the B cells in human anatomy. So, cellulose is kind of the principal component of the plant cell walls. It's made up of repeating monomers of glucose attached end to end, so it's a long chain. And microfibrils are kind of bundles of cellulose, so about 10 to 25 nanometers in diameter. And they wind together to form fine threads that wind together kind of like cable strands and are stronger than steel would be at that thickness. Then we've got hemicellulose, that there's a lot of variation for those, but they're hydrogen bonded to cellulose microfibrils and act as tethers and limit the potential expansion of the cell wall. So it's kind of like Right. If you're in a building, shall we say? Yeah. Stronger than steel. If steel and the microfibrils were the same thickness, they would be stronger than steel. But let's say you're in an old building and okay. Let's say you're in an old building in San Francisco, right? Because it's notorious for earthquakes and everything else. And it's an older building, so it needed to be retrofit. It's kind of a warehouse or some kind of large structure, church or something, that has kind of a vaulted ceiling. Because those tend to fall more than if there's a lot of interior beams and whatnot. The hemicellulose are the retrofitting reinforcement cables that they would put up on the interior as a reinforcement support to make sure that the walls are all stable. And if something happens, the base structure isn't going to crumble. It's not going to disrupt the function of that building, that cell that's being worked with. And thus the cell wall is stabilized because it has something to lock it, essentially. And we've got the pectins that are part of the first formed or primary cell wall layers. And the intercellular substance, so the middle lamella that, okay, so you can see it in here. It's the bit between the cell walls, essentially. And cements contiguous cells together. So the cells that are up against each other, it cements them together. It's hydrophilic. It's made of hydrophilic polysaccharides that attract water-bearing pliable properties to the wall, allowing it to expand. And that kind of relaxes the static structures that are present in the plant cell walls and allows it to grow more as needed. It also adds the kind of gel texture to jams and jellies and fruit preserves and stuff like that. Then we've got glycoproteins that are structural proteins for the cells. And one of the big ones for that is extensins, which, let me just say, as someone who has dealt with speech issues their entire life, that is a really hard word to say because it sounds like I have a weirdly specific lisp for extension, but it's not. And it's a completely different word. It's messing with my head a little bit. So, glycoproteins, they're hydroxyproline-rich proteins, and were originally presumed to be kind of involved in the cell wall's extensibility, so the ability to extend. But more recent analysis has kind of concluded that it might just strengthen the cell wall instead, thus limiting extension. So it's now been relegated to the it's called Greenland and it's not green sort of thing. And then we've got lignin that is also in that same category, that adds strength and bending stiffness, which is a weird phrase because apparently bending stiffness and rigidity are the same thing, so why not just say it adds rigidity to the cell walls? I don't know. So, there we go. And then we've got cutin, suberin, and waxes that are all fatty substances commonly found in the walls of the outer protective tissues of the plant body. So there's a lot of things in cells kind of in the interior, but if you go to the exterior where it's like the surface of the leaf or the surface of the stems, around the base of the flowers, that sort of thing. That's where we're going with this. So, cutin, for example, is found in the walls of the epidermis, so the very outside of the cell. Very outside of the plant, like the top layer. Just like for humans, the epidermis is like this right here. I realize that you can't see anything and my hand's in the way anyway, but the epidermis is just the top layer of your skin. That's what you're feeling if you touch your arm. The severin is found in the secondary protective tissue, like the cork. That's in a lot of trees and things. It's in other element, like other parts of plants too, but the most notable is like the outer bark of the tree a lot of times. In the primary wall, the cellulose layers that formed first, hence primary. The middle lamella is the space between the primary and secondary cell wall, also called the intercellular substance. Yeah. The secondary cell wall is from, like, the cells sometimes deposit additional wall layers, and if that occurs, it's laid down by the protoplast on the inner surface of the primary wall. And something to note is actively dividing cells only have primary cell walls. They don't have the secondary. The primary pit fields are thin areas in the cell wall, and plasmodesmata, the fibrils, commonly aggregate in pit fields but are not restricted to them. Allows for easier transmission between cells. And again, that'd be these pit guys around here. The secondary wall has pits in places with no primary pit fields, and cellulose is more abundant in the secondary walls, and is only deposited or forms when the primary wall growth stops. So when the cell has reached the size that it is going to be, then the secondary wall is built up. And then when it's built up, all the other structures that make sure that it doesn't move, doesn't go anywhere, doesn't expand anymore. And that's also part of why really young plants, the seedlings and saplings and stuff, are a lot more prone to having issues if they're over or underwatered, because they don't have that base protection that's inherent to plants. Like, they have some level of it, but they don't have the fully developed tough cell wall structure that prevents too much or too little water being in the cells. And without that, it's absorbed at a higher rate or it's released at a higher rate than might be needed. So just keep that in mind. Just like with tiny humans, tiny plants need extra care. That's why they're in a nursery sometimes. Alright. So, there's a few just quick bullet points, and then we'll get into the last bit. Or, no, the next bit. So, pit membrane between two pits. Kind of self-explanatory. A pit pair would be two opposite pits and the membrane. So, like, if there's a pit here, and there's a pit here, and the membrane between them. Simple pits, there's no, like, overarching wall or anything. It's just a pit. For bordered pits, there's a secondary wall that arches over the pit cavity. Cellulose synthase would be a synthesizer for cellulose microfibrils that occurs in the plasma membrane. Yeah. And in plants, they appear as rings or rosettes of 6 hexagonally arranged particles spanning the membrane. And got protoplasts that are it's like okay. Got protoplasts of adjacent plant cells that are connected with one another by plasmodesmata. And most are formed during cell division. Okay, now the main meat and potatoes, as it were, of the chapter. Because, all the rest of it's just been lead up, I realize. But now we've got the cell cycle. And as part of the cell cycle, cell division. So, cell division, kind of self-explanatory, the process by which cells reproduce. They divide. Daughter cells are the resultant cells upon the completion of cell division. Each inherits about half the cytoplasm from the parent cell. It's pretty much an even split, and is genetically identical to the other daughter cell and the parent cell, because the genetic material didn't have anything to combine with it, it just split in half. Alright. So, we've got cell division is broken up into 2 parts. Mitosis, that's nuclear division, so the nucleus dividing, and these are occurring at the same time most of the time. So, nuclear division. It's the nucleus itself dividing. And it's a complete set of previously duplicated chromosomes is allocated to each of the 2 daughter cells' nuclei. So the nucleus is over here, it replicated its contents and is splitting. Meanwhile, there's cytokinesis, where it's a process that divides the entire cell into new cells. So each new cell contains not only the nucleus with the full chromosome components, but also about half of the parent cell cytoplasm and all the other bits in there. So the nuclei separate off, and the rest of the cell separates. We've got interphase. We have an intense cellular prep sort of situation wherein the cell is getting ready to divide, including chromosome duplication. That's kind of it's usually illustrated as G1, S, G2. So, phase 1, status, phase 2. The cell cycle itself is generally termed or generally described as mitosis and interphase. That's the nuclear division and the preparation for division. M phase, that's mitosis and cytokinesis. And yeah, and then it repeats, essentially. Then there's initials that make up the apical meristems that are embryonic tissue regions, essentially. That's the generally topmost point and bottommost point of the plant. And in addition to the other phases, there's also the G0 phase that is essentially the plant going dormant. Usually it's in the winter, but sometimes it's the summer. Just depends on whenever that plant goes dormant. And there's checkpoints. And for that, it's transition points in the cell cycle near the end of G1 and G2 that either the cycle can stop or to initiate, stopping. The cycle can either stop and initiate phase S, that's just stop, or wait. I read that wrong. I might be able to tell. This part is not my expertise. Okay, yeah. Okay, so for the checkpoints, the plant or the cell has the option of either stopping the cycle and just static, or initiating S phase and starting another round of cell division, because S phase is synthesis. It's synthesizing more cells. And we've got the centrioles that are cytoplasmic organelles found outside the nuclear envelope and are identical in structure to the base body. They're found in cells of most eukaryotes aside from fungi, red algae, and non-flagellated plant cells. And they divide and organize spindle fibers during mitosis and meiosis, surrounded by centrosomes. Yeah. So there's that. The primary role of G2 phase is to check the DNA for any abnormalities or damage and make sure that it is repaired if possible. There's also the phragmosomes that contain both microtubules and actin filaments and transverse, and then what was I writing, honestly? Yeah, okay. Which is a transverse sheet of cytoplasm. And we've got the pre-phase band, which before the phase. So it's before mitosis. And that's when the spindle that is going to pull apart the chromosomes and whatnot is forming. The cell plate is the initial partition between the daughter cells. So the nuclei and everything are pulling apart, and there's a cell plate that starts forming between them to block off what goes into which side. That's the initial separation point. Okay. So, one sec. Gotta resituate over here. Okay. Anyone have questions so far? Kind of a lot, I realize. Awesome. Okay. All right. Alright, so now mitosis and cytokinesis. Alright, so prophase is the first phase of mitosis. It's the name, prophase. But it's not a clearly sort of defined event. It's just kind of a amorphous sort of thing. The chromatin, which is kind of floating around in the regular non-dividing nucleus sort of coalesces to define into defined chromosomes, and then it's replicated. So the chromatin is the base element of the chromosomes, essentially. And during the S phase, so the synthesis phase, the chromosomes duplicate, right? Hence, each chromosome consists of 2 identical sister chromatids. Then we've got the centromere, that is the midpoint on the chromosomes. It's a constricting sort of spot on there. Centromeres consist of specific DNA sequences needed to bind the chromosomes to the meiotic spindle, which forms during metaphase, which is the next phase of mitosis. And metaphase begins as the meiotic spindle, which... Oh, what are you doing? Okay. Begins as the meiotic spindle. It's kind of a 3-dimensional structure that's the widest in the middle and tapers towards the poles. Appears in the area formerly occupied by the nucleus. So it's got some bulk here. It's tapering to the ends. So kind of think like a real squidgy football sort of thing. We've got quintotors. Very strange word. Oh, come on. That are specialized protein complexes that become attached to or capture microtubules with the sudden breakdown of the nuclear envelope as division is starting to happen. The shortest phase of the whole thing is anaphase, which begins with the simultaneous separation of all the sister chromatids at the centromeres. So all the chromatids separate and are now daughter chromosomes. Okay. And we've got motor proteins like dynin or kinesin, so kinase kinesiology movement, that use ATP and pull the chromosomes along the microtubules to the poles that they're supposed to go to. Well, tubulin subunits are simultaneously lost at the kinetochore. Again, science, weird words. During the telophase, it's the next one, there's separation of the two identical sets of chromosomes, or that's when the separation is completed, rather. It's completed as the nuclear envelopes are being organized around each set. So it's a preliminary envelope to encase the new, what would become a nucleus, essentially. Okay. And depending on the cells and the conditions and everything else, different stages can last longer or shorter. Different elements can happen or not happen, but prophase is always the longest and anaphase is always the shortest, regardless of anything else. And cytokinesis, as the name kind of suggests, is the division of the cytoplasm. So the squidgy bits in the cell, essentially. That typically follows mitosis. Okay, so then we've got the next bits. So in early telophase, so there's an initially barrel-shaped sort of system of microtubules called phragmoplasts. That the phragmoplast forms between the two daughter nuclei. So the nucleus has split, but the rest of the cell is still fairly together. And the phragmoplast, like the meiotic spindle that preceded it, because it's essentially the spindle that has been cut in half by the cell plate. It's composed of overlapping microtubules that form two opposing arrays on either side of the division plate. Because it's basically like there's a long cable, like forcing 2 points to opposite sides, right? Let's go with this. Yeah. All right. Here we go. Yeah. We'll go with that. All right. So the spindle is a whole bunch of these. And we've got one daughter cell over here, or the daughter nucleus is over here, and one is over here, and the cell plate forms right in the middle. So these are still there, but they're separate. They're separated now. So these become the phragmoplasts, and then are incorporated into the daughter cells as microtubules. Okay. And then the developing cell plate fuses with the parent cell wall at the position previously occupied by the prephase band. So the cell plate starts in the middle and just kind of continues out. Like, if at some point you play Jezebel, it's the same principle. Because for that, if you say you click in the middle of a section, it'll send out part of a wall on either side. It's the same principle. And that blocks the balls from coming into the other sections, and you can wall it off and stuff. But it forms a median cell wall, and each of the daughter cells in the interior of the previous cell have their own cell wall, and then those eventually stretch more and form like the main cell wall, and is reinforced as such. So yeah. Ow. Alright. So I don't know how well this is gonna show up, but oh, come on. Yeah, that's not showing up for anything really. But essentially, oh jeez. Yeah, I'm really thinking I need a better webcam than this guy. Oh. Alright. So there's that. So that concludes chapter 3. The next chapter before, and it's the movement of substances into and out of cells. Alright. Anyone have questions or commentary or what have you on that sort of thing? Or not? Nah. Alright. Well. Hmm. Uh. That is a question. I mean, if she was small enough, probably. They're probably kind of. They seem to be kind of blunt, but they have some force behind them, so, like, I could see it happening. Mm. Alright. So, with that, um, again, if you want to be notified. I mean, that's fine. They're always welcome to have fun with the plant stuff. I was trying to figure if there was a typo in there or if it was like a funny, and yeah, yeah, that's all. Uh, but yeah, if if they're strong enough to have an equivalent that's higher than steel, you could definitely do some damage with that if the individual being stabbed was small enough that it had some kind of effect. Because, I mean, like, bamboo works. I don't know. That would be something interesting to look into. I honestly don't know. Uh, but be interesting. So I know spider silk. I've heard spider silk more described in terms of Kevlar than steel, usually. But that might just be because I've watched so many sci-fi movies that are kind of like weird B grade, right? At this point. So it's kind of skewed the data input on that one. But they're probably kind of comparable, really. Uh, if you, especially depending on the plant, Just a little bit. Just a little bit. I can't remember what it's called right now, but there's one that it was like stupid bad, but it was hilarious at the same time, where there's termites from space that have taken over Oregon. Yeah. With Bruce Campbell as the lead. It's so bad. But I would say they're probably kind of comparable. For that, because if you try and it really is, if you try and just tear, it's plugged in. Why are you? Okay. Apparently, I have to stop the stream now anyway, because even though it's plugged in over there, my computer's saying that the battery's too low and it's gonna die. So, I'll leave you with that. And Staring will talk to you in a little bit. But if you'd like to be notified when I post stuff, there's a few things that are in the works that just need to get edited, and a few others that need to get filmed and whatnot. Yeah, really. Hit the subscribe button down below and the bell icon next to it. If you liked the video, leave a like on it. I would appreciate it. And I will see you next time. All the links to everything, including the textbook, if you want your own copy, are in the description down below. And there's a link to the playlist for all of the chapter breakdowns in there, and in the iCard, whichever corner it is over there, I think. So, yeah. Until next time, I will see y'all later, and have a good one. There should be, if I can get it written, there should be an episode of the podcast out tomorrow, too. So, we'll see. Alright. Till next time, I will see y'all later. Have a good one, everybody. Stay safe. Bye.